Many people have asked the question lately over the past few months or made comments on social media, on YouTube, on Facebook, as to what is the best ingredient to support the natural release of stem cells from the bone marrow. And I thought that the best way to answer these questions is simply to go back to the data, to the scientific data, to what has been published in the scientific literature, in the peer-reviewed scientific literature. And it starts with the very first ingredient that was documented to support the natural release of stem cells from the bone marrow, which is steminins. Actually, it's the work that was done on steminins that really led to the development of what is today known as the natural repair system, the natural renewal system that is based on the release of our own stem cells. So if we go back to that paper, which was published in 2007, we can see that the median release of stem cells, or the median increase in the number of circulating stem cells after 1 hour, is roughly around 50%. This was also confirmed by a third-party study that was published in Egypt, I believe it was 2013, when the exact same increase in the number of stem cell in circulation was documented 1 hour after consumption of steminins. And what I want to point out here is that over the years we have talked about a 30% increase in the number of stem cells in circulation. And the reason why we said this is simply because in the early days, when all of this documentation and all these studies were done, and we were getting ready to bring steminins to the marketplace, our legal counsel at Desert Lake Technologies were telling us, you know, stem cells is linked to diseases. So if you make a stem cell claim, you'll probably be in trouble with the FDA. And at the time, what I was saying is that no, we will not be in trouble because it's a function of the body. So this is going to be a functional claim. But we were into this argument with our legal counsel, and finally the compromise was to make a statement about stem cell support, but to keep it very, very conservative, to keep it as low as we could, to make it significant but really not too high. So we basically talked about a 25 to 30% increase in the number of circulating stem cells. But if we go back to the data that was published, it's a median release of 50%, and in one individual we gave that person 16 different times, 16 different days. We gave him steminins, and then documented the increase in the number of circulating stem cells. And the range here is pretty wide, going as high as 325% increase in the number of circulating stem cells. So that's what we have with steminins. From there, we started to look at other ingredients, and we looked at ficoidan from Undaria pinnatifida. And what we found was that after the consumption of Undaria pinnatifida, the ficoidan from Undaria pinnatifida, we have an increase in the number of circulating stem cells of about 18%, which is much less than what we have with steminins. But what was very interesting here is that that release was sustained over time. So the idea was that if we can blend these two together, we are going to get a very interesting synergy. And that's what we got. We got a very interesting synergy in terms of a significant release of stem cells from the bone marrow that was sustained over time. So the line that we see here, the curve, is the combination, the blend of these two ingredients. So some competitors on the marketplace are using this curve to say that that was a response to ficoidan. It is not. It is not accurate. That was the response by combining both steminins and ficoidan. Then another ingredient that was discovered to trigger stem cell release from the bone marrow was allomacroclada. Again, here some exaggerated statements and claims have been made. The best is to simply go back to the data that was published just a few years ago. And the data is showing that within a few hours of consuming allomacroclada, we have an increase very comparable to what we have with steminins. Again, it's sustained over time, but what was really the discovery, if I can say, with allomacroclada is that it increased the number of one specific type of stem cells called endothelial progenitor cells. These are stem cells that lead to the formation of new capillaries, and it's something that is very important in the body to deliver cells, to deliver nutrients, oxygen to various tissues. So if you can have better circulation, then obviously the entire body can function better. So that's the secret behind these type of stem cells called endothelial progenitor cells. So allomacroclada led to that increase. Now again, it's by blending the three together, steminins, ficoidan from Undaria pinnatifida, and allomacroclada, that we get a very interesting response. But here again, some competitors out there on the marketplace are using this curve, which is the combination of all these ingredients, to claim that this was the response to allomacroclada. Again, this is false and inaccurate. It is the response of all of these three combined together. And the last ingredient that was documented is mesenchyme. Mesenchyme is an extract from spirulina that was developed and discovered as Cerule was developing Cyactiv, which is a concentration of phycocyanin from spirulina. So this yellow liquid called mesenchyme was discovered along the way, and then we started to do some work on it and discovered some very, very interesting things. Mesenchyme, within an hour of consumption, will increase the amount of GCSF in the bloodstream. Now, GCSF is the natural signal that the body sends to all the to the bone marrow to trigger the release of stem cells from the bone marrow, so that these stem cells can go and repair various parts of the body. So what mesenchyme does is to really tap in the very signal that damaged tissue in the body will trigger to call for stem cell release from the bone marrow. And as we looked at stem cells in the bloodstream, we saw that mesenchyme also increases the number of endothelial progenitor cells up to 60%. All of this is in preparation for publication, but it is to show that mesenchyme gives us a response very comparable to what allomacroclada was giving us. And when we blend all of these together, this is where we get, again, the best response. When we blend Steminence, Fucoidan from Anderia pinnatifida, and mesenchyme. So I think the take-home message with all of this is that all these ingredients are very interesting ingredients. It's very difficult to say which one is best than the other, better than the other. I think the real take-home message is to understand that it's when we blend all of these together that we really get the strongest response. And at this point in time, in terms of actual products available, Steminence Ultra is really the most powerful product, because it does blend Steminence, the original ingredient, Fucoidan from Anderia pinnatifida, and mesenchyme that has an effect on GCSF and endothelial progenitor cells. So the take-home message, blends of these ingredients is really what is the strongest product to take to support the natural release of stem cells from the bone marrow. Thank you.